Oui, oui, quelques minutes, enfin quelques secondes. C'est bon.
Merci.
Quelques minutes.
So, good afternoon or good morning, everyone. It's my pleasure today to introduce Alex and Moksh. They are both working at Mila. Alex is a postdoctoral researcher there, and he got his PhD in 2020 from the University of Osnabrück in Germany. And his current focus is on machine learning to fight climate change, mostly by accelerating material discovery. And Moksh is a PhD student in the University of Montreal and Mila. And his current focus is on deep learning and in particular on active learning. And both are closely working with Professor Joshua Benjo. And today they are going to tell us about GFLOWNET, which is a recent and very interesting method that appeared in the literature. And they are going to provide us a theoretical introduction and overview of the method, as well as applications onto biological sequence design. So thanks for accepting the invitation. It's a pleasure to have you here, and we are very much looking forward to your presentation.
Thanks for the introduction and thanks for the invitation. So yeah, let's get started. So today we'll be talking about like a friendly introduction to GFLOWNET, and so that we can tell the world about this cool new method that we have come up here. And how we leverage it for doing biological sequence design. Yeah, so before I get started, I should probably note that like the work we'll be presenting here is sort of a collaborative effort with a bunch of people here at Mila and at other institutions, and we have had like a bunch of papers on GFLOWNETs, which you can find here. And so hopefully we'll be able to give you a flavor of the method today and like why we are so excited about it. So one of the key motivations that we had like when we were sort of coming up with GFLOWNETs was this particular problem of drug discovery. So the drug discovery problem is generally like a loop of like design and evaluation where you have like essentially a generation process where you generate a bunch of candidates, and you have some evaluation where you black box, let's say, where you can evaluate the usefulness of a particular candidate. So for instance, let's consider the problem of like discovering small molecules which can sort of bind to ligands to stop their activity. Ideally, like what we would want is we would want to discover small molecules, like drug-like small molecules, which can actually achieve this within humans. So we administer this particular small molecule to humans, and it will sort of like reduce the activity of a particular disease ligand and so on. The tradition, like, so this is not necessarily like a very linear two-step process. It's a bunch of like design and refinement, and this traditionally involves things like in silico screening, where you go through like a bunch of known molecules, you screen through them, or you have some simulators which can simulate certain properties of your like desired effect. So for example, if you want like if you want to just stop the activity of a particular like virus, let's say, for example, you would have a simulator which sort of simulates the biophysics of the interaction of molecules and this particular virus. Another thing would be in vitro screening, where you like these days there are like methods which can do thousands of experiments, like synthesize thousands of proteins and test them against a particular target. And there would be a few rounds of these smaller tests, and then you would move on to something like testing in mice, and then eventually human trials. So another key aspect to note is like when we go from, let's say, in silico screening to in vitro screening, you have to screen through a large number of candidates, in the sense that from a search space of like 10 to the power 10, which you could do probably in silico screening, which is like around tens of billions of molecules at a time. You can't do tens of billions of experiments. So you can only select, so you only need to select the most promising few to actually take into the next round. So the key idea here is that when we are doing in silico screening, the scoring functions that we use, so something like a docking simulator, which simulates how a molecule binds to a particular ligand, these are often unreliable, and they can, in many scenarios, not be very correlated to the final desired property that we care about, which is activity or like safe activity within humans. So one of the main aspects is that like it cannot capture all the properties. So for example, a docking simulator cannot actually model things like how it would interact with the systems in a human body, because some of these things might not be specifiable itself. And another thing is there are lots of approximations made in these programs and simulations to make them actually like sort of run quickly on a computer. So these approximations also make the scoring function very unreliable, in the sense that if you get a very high score in these simulators, it might not really translate into the actual desired effect in the real world. So traditional methods which sort of do these in silico screenings, they focus a lot on finding like a single best candidate, which can, let's say, maximize the scoring function. But what these techniques miss out on is that since the scoring function is unreliable, you don't necessarily want just a single maximum. You don't want to put all your money in a single basket. You want to essentially capture the modes of your reward function, in the sense that you want a set of molecules or your set of candidates which is very diverse. So for example, even if your scoring function is just like one of the properties which you would like to capture, if you consider the modes of modes of your reward function, you would also get like good enough molecules for the scoring function, which might actually end up being better in like the next steps of your drug discovery process. And another reason why the diversity in these batches is important is because the mechanisms with which these candidates interact with your target, they can be different. So for example, a particular virus can have different target mechanisms in different, let's say, temperature regions, in different like environmental conditions and so on. So it is like, even from that perspective, it is important for us, because there is no one size fits all, even in like drugs. So it's important to capture like a wide variety of these things, so that we can eventually get something which is useful. Yeah, so now that I've like sort of motivated this problem that we have at hand of like discovering like large batches of diverse candidates, so why do we need GFLOWNETs? Like, before I tell you about GFLOWNETs, let's think about what current machine learning methods already offer us. So one of the sort of very clear and like clearly useful methods is MCMC methods. So MCMC methods have like traditionally been like a very like strong methods for like getting samples from a particular unknown target distribution, and they have powered a lot of innovation in like the Bayesian inference community, for example. But one of the sort of fundamental limitations of MCMC methods is there are two fundamental limitations in the context of the problem that we're trying to solve. One is that the mixing times in your MCMC methods, they can be exponential if your modes are well separated. So just for instance, if you look at this particular example, we have like fairly well separated modes, and MCMC tends to just sort of not be able to move from one mode to the other. Now, of course, like given infinite time and compute, this will eventually cover the entire distribution. But the problem here is like in cases of high dimensional distributions where you do not have infinite compute, this can be a limitation. And another important aspect here is also that sampling from MCMC is sort of the compute is towards the compute happens during sampling. So if you want to generate a large number of batches, you have to spend a lot of compute like during sampling. So that's one of the drawbacks of MCMC methods. Another possible candidate one might think is reinforcement learning, but with some exploration. So like I mentioned earlier itself, which is that maximization is not necessarily what we want. We don't want like a single maximum, like single best molecule that we want to generate, but we want to capture a sort of diverse set of molecules which are all good. So reinforcement learning with some appropriate exploration can seem like a very appealing method in these settings. But again, so traditional reinforcement learning, the objective itself is to capture like to learn a policy which will get you the highest reward. So it already focuses on getting the highest mode of the reward rather than capturing all the modes, which can be a problem. So one can also think that, okay, how about I have like a very efficient exploration method? But the thing is, efficient exploration in RL is also another like open research problem, which we do not have methods for. And there is also another more technical issue in this particular setting where you are sort of constructing molecules using reinforcement learning, which is if you have multiple ways of multiple ways of constructing one particular state. So let's say multiple trajectories leading to a particular state, that state will get its value overcounted, and just because it has like multiple paths leading towards it. So like we have a lot more discussion about this in the paper, but I will just skip over it for now. But yeah, so these are like some challenges. So this is sort of what like motivated the need for like a new method, but which combines a lot of aspects from MCMC as well as reinforcement learning. So yeah, this is what we call our generative flow networks. So let me set up some preliminaries here, which is what are we sort of what is the setting that we are sort of working in. So we are for the purposes of this talk, we'll be focusing on settings where we are trying to sample or where we're trying to construct a discrete object. So in this example, let's say this Lego car. So that's a discrete object of interest, and this is like a compositional object in the sense that we can construct X by putting together a bunch of building blocks. So in this so for example, you can see in the image, you have like a bunch of Lego blocks, and you can like put them together in a particular way to get your car. Now note that like there can be multiple trajectories which lead to this end product of getting a car, which is you can like change the steps. Some of the steps can be sort of interchanged so that you will have like multiple different ways of reaching that object. And yeah, so at each step you will have a partially constructed object, which is like let's say, for example, the second step in the figure, which is you just have like a base of the car, for example. So after adding each block, you will have some partial object. So this essentially is what forms like our state space, which is your partial objects, and your like set of fully constructed objects would be a subset of this state space of like all possible objects using those blocks. And we also assume that there is this special starting state, which is empty, where you have like added nothing to your object. Where you have added no building blocks to your object. And we have this final state SF, which is where all the trajectories end at that particular state. So you can think of it as like a done action. So, okay, I'm done now. That's done generating the object. Yeah, so this is the general setting. Now, this process of constructing an object using a set of building blocks with like partial objects as intermediates, it induces this directed graph where the set of where the set of nodes S is essentially your state space of basically all the partial objects that you can construct, and these edges, these objects are connected by edges when you can reach from one object, where basically you can add a particular action to this partial object to reach this other object S prime, let's say. So this graph essentially will cover all the possible configurations that you can put together given your set of blocks. Now, there is one assumption which we make here, which is that these actions are all constructive in the sense that you cannot, let's say, delete a block and then sort of add it again. So this will basically introduce cycles in this graph, because you'll be able to remove an object, and then, sorry, remove a block and then add that block again. So we assume that the actions are only constructive and you can just keep adding blocks and you are not allowed to remove blocks. So it's a DAG, yeah. So it's a directed acyclic graph. Now, so what GFLOWNET's learned to do here is essentially just learn a policy pi to construct this object through a set of through a series of actions, such that you sample an object X from this policy proportional to the reward for X. So the reward for X, so if you are constructing, if you are considering this problem of constructing a Lego block or this Lego car. So this Lego car is, so let's say our reward would be, if it is a vehicle, then I give it a 1, or if it's not a vehicle, then I give it a 0. So that could be the reward. And I want to generate based on this reward. So I will tend to generate like things with higher rewards. I will tend to generate vehicles. But it will not be fixed to just one vehicle. It can be like, you can have multiple vehicles or multiple types of vehicles you can construct using the same set of blocks. So one thing to note here is that the way we learn GFLOWNETs, contrary to what I mentioned for MCMC methods, where computation happens during sampling, here we amortize the computation during training. So we do a bunch of these constructions and learn from these constructions. And during sampling, we just have to run the policy, which is much cheaper. So the computation is amortized, which also gives us sort of a computational advantage over MCMC methods. Yeah, so now to delve in a bit deeper into like what actually happens within the GFLOWNETs. So first we introduce this notion of flows. So like, I guess before I even explain what the flows are, I should note that these are not the same as normalizing flows, because this is a question we always tend to receive and people tend to confuse the methods because of the name. But yeah, these are not normalizing flows. So these flows are more analogous, or like these flows are essentially the flows from graph theory, like the flow network flows, where you have a weighted graph and you like the weights of this graph are essentially your flows. So the setup we have here is essentially of like a flow network from graph theory, which is you have some sink state, you have some source state, you have a flow, like some quantity of flow from your sink state, and everything starting from the sink state, and everything ends up in the source state. Sorry, everything starts from the source states and ends up in a sink state. I said this the other way around. So yeah, this is the graph theoretic flows. Now, to like one way to sort of intuitively think about them is you have let's say network of pipes, and you have like water coming in from one particular source, let's say a lake. So from this source, you'll have some water which flows through all of the pipes, and the how much water flows through each pipe is sort of the flow of that pipe. Now, the way we define, so this way we can sort of define a few quantities on this graph that we talked about before. So one of them would be the trajectory flow. So if you just take random walks on this graph, starting from the empty state to any like to the final state, this random walk will form a trajectory, or what we call a trajectory. And this trajectory, so we define this trajectory flow, which is essentially how much mass flows through this particular quantity. Through this particular trajectory. Then we define what is called a state flow. So the state flow would be essentially how much flow passes through this particular state. So like all the trajectories which pass through a state would contribute to this state's flow. And the edge flow is essentially just the smallest quantity, which is like at a particular edge, how much mass is flowing through that edge. And using these flows, we can like define some policies. Essentially, this policies is what these policies define is that we act based on the flow. So if the flow is high through one particular edge, we would select that edge, essentially starting from a particular state. And there is a backward policy here as well, which is, so this policy, so there are two policies. One is where we go from the empty state where it's sort of constructive, and we have this other policy which is sort of learns to go backwards given a particular object. So like we sort of learn how to deconstruct this particular object. Like this will sort of become a bit more clear when I go to the when I talk about the learning objectives. But yeah, so essentially these flows are you can think of it as like how the probability mass flows through this graph that we have defined. Yeah, now coming to the this is essentially like the key result which underpins all of GFLOWNETs. First I define what is called a consistent flow. So a consistent flow is essentially a flow which sort of a flow which is conserved. So how much ever flow I start with at the source node ends up at the sink node. So S0, you can think of it as your source node, and SF is the sink node. So whatever flow starts from S0, all of it ends up at SF, which is your sink node. And we also assume that this is true at each and every node in your graph. So at each node, whatever is the incoming flow, it is equal to the outgoing flow from that node. So essentially what we're saying here is there is no loss of flows. Essentially there is no loss of this mass that we are sort of propagating to this graph. And at each node or each state, the flow is conserved.
It's like energy conservation.
Yeah, it's like the principle of energy conservation and like the broader principle of conservation, which appears in a lot of things. So yeah, so the consistent flow. So yeah, that's how we define a consistent flow. Now the key result is that if we have a consistent flow where the flow at the terminal state, so the terminal states are essentially like our completed objects, like the final objects in the trajectory. And if we set those flows to the reward of those particular objects, then the policy which we defined earlier, the forward policy, it will sample or it will construct an object X proportional to its reward. So if the reward is high, it will be sampled more often. If the reward is low, it will be sampled less often. So this is essentially the key result, which is that we want to learn if we are given a consistent flow, then we can sample exactly from the distribution induced by this reward function. So another assumption I should also mention is the reward function is assumed to be like non-negative, since we are like using it as like a density function of a distribution. But yeah, so this is like a very interesting result in the sense that if you satisfy this very simple conservation property, you get this like amazing result that you can sample proportional to your rewards. And another interesting part, which is like a very like sort of well-studied problem, is like learning this log partition function, or the partition function for a distribution, which is induced by the reward function. In GFLOWNETs, it is essentially the flow at your sink node, at your source node. So if you know if you just compute the flow at your source node, it will give you the partition function of your distribution. So this is really interesting because people have tried to like come up with a lot of methods to estimate like the partition function of distributions. And here, by sort of leveraging this compositionality in the space and like the power of like neural networks which can generalize, we can learn this partition function like fairly easily. Yeah, so now the question comes is, how do we learn these flows once we know that we can have these really nice policies which we can learn from, we can define using these flows. So yeah, so like if we just look at this flow consistency, so essentially the goal here is we want to learn this function, this flow function f, which is sort of generally in our case parameterized with a neural network. So we want to learn this flow such that we satisfy this flow consistency equation. Now we can very simply just take this condition that we want to satisfy, and we can define a TD like objective using this constraint. So essentially we try to make the flows at each state consistent. So this gives us this flow matching objective, which is essentially very similar to a TD objective, just in the log domain. We use, so another note here is we use the log domain since the flow quantities can be like, sorry, the magnitude of the flows can be wildly different. So that might cause numerical issues, but yeah, like it also works if you just remove the log. But essentially here, this flow matching objective is just taking this constraint as it is, and then just defining a temporal difference objective using that particular constraint. And another more recent learning objective that we can define is this trajectory balance. So it essentially stems from another result, which we are not covering here, about the detailed balance condition, which is very similar to the flow matching condition, which is sort of shown here. But this is an objective which works much better in practice. It has better credit assignment properties. We won't go into much details about this here, but please, you can take a look at the paper. But yeah, just to summarize, essentially, you have this method which works like a reinforcement learning method. So you sample trajectories and then you apply this learning objective and you're good to go. Surprisingly, we can learn learn to sample from very complex distributions using this. So now Alex will talk about how we leverage this for active learning.
Yeah. Is there any clarification questions maybe before we can delve into these details?
I think you can go ahead and.
All right.
In case.
Just wanted to make sure that the theoretical part is understood. So yeah, I'm gonna guide you in the next few slides through how we can, an application of how we can leverage the properties of GFLOWNET for improving active learning methods. Before I go into that, I want to motivate the need for such a method by saying a few words about what the traditional pipeline for discovering biological sequences in this case, but in general for scientific discovery in other domains. So typically you need a human in the loop that leverages their expertise to come up with queries about a particular problem. So for example, if we are thinking of proteins or DNA sequences or whatever, the human is the person who says, okay, this might be a good candidate. And then you have to test it in the real world. That means either a computationally expensive simulator like molecular dynamics, or it can be even a wet lab experiment like Mox explained in the first slides, like animal testing and so on. This is, of course, very slow and expensive, but you end up having some results, not many. Then you learn from these results and repeat the process again. So this is, of course, slow, time-consuming, financially and computationally expensive, and relies very much on human expertise. Something we can do with machine learning is replacing the real-world oracle, so the wet lab experiments or the expensive simulator. We can replace this with a model that we train, a machine learning model that we train with the available data. And this already improves things a lot, because then we might jump from very costly simulations to models that can run in a few minutes, hours, days, but still, even if it's long, it will be a lot faster than the real world simulations. This is great, but of course it introduces noise, so there is a cost in it. It won't give you the same accuracy as the real world models. And another thing to note is that even if it's a great improvement, this only provides linear gains. So if we were able to do our experiments in, I don't know, weeks with the real world experiments, now we do it in some days. That's good, but whenever we are exploring, if we are exploring like very large search spaces with a lot of candidates, this is likely not to be enough. And this is where GFLOWNET gets into the game. So we can improve this. We can do better than linear gains by introducing an agent in the loop. So this part, so that I replace the scientist with an agent. So in this case, because we have access to first we have access to real world data. We use it to train a machine learning model, and now we can use this machine learning model as a proxy as an oracle for our agent, and the agent is able to learn from the outputs of the oracle, and ideally learn the structure of the search space. And if there is indeed structure in the search space, what Moksha was describing before, then it is possible to actually generalize, or ideally, theoretically, it should be possible to generalize this structure to allow the agent to explore new regions of the search space. And this leads to the key result here, or the key message here, which is to construct better queries that then we can submit to the costly experiments, but with better confidence, get new data, and retrain our models, and so on. So this is the typical active learning pipeline that many of you might be familiar with. The key here is that with an agent that can learn structure from the data, we can construct better queries. And this would lead us to exponential gains in this active learning game. Right, so in the next few slides, I'm gonna be even more concrete and talk about a particular case, a particular application, which is the discovery or design of antimicrobial peptides. So peptides are short chains of amino acids, like proteins. And the goal here is to discover peptides that have antimicrobial properties. For this, there are data available. For example, I'm giving the reference at the bottom. So we have some data, the initial data set that I was mentioning before, and then we can run active learning on this. In this case, we run 10 rounds of the whole active learning pipeline with a batch size of 1,000. So that means that every round we are generating data from our proxy model, from the machine learning model, and selecting 1,000 data point sequences that we add to our initial data set and run it again. Okay. In this case, this data can be conceptualized as chains. So these are sequences, chains of length up to 50, or they can be shorter in our experiments, and they are formed with a vocabulary of 20 amino acids. Okay, so that yields about 10 to the 65, which is huge, as you see. So it's not a trivial problem to find good candidates here. Now, before showing the results, what do we want from the results? So this is reiterating on the main idea about the motivation for GFLOWNET, which is that it is not enough to just have a good performance. We really look for diversity and novelty. So of course we want a good performance of the best set of candidates. In this case, we will focus on the top K. K can be maybe like 10 or 100. We will show results with 100 later. We definitely want that these 100 best candidates have a good score. Need to discuss the details of the score, but the score will be like a metric of the goodness of the samples. But we also want them to be diverse for the reasons that Mox described at the beginning. There might be noise in the oracle. We really want to explore different sequences and so on. And of course, this is sort of obvious, but it's important to mention novelty is important. So we want to discover sequences that were not included in the original dataset. So the precise way we define this metrics is in the paper. So as a conclusion from this slide, a set of candidates should be evaluated holistically. That means all the three metrics are important. It's not enough to have just diversity, but then bad score. It's not enough to have good score, but bad diversity and so on. So in the paper, we compared training GFLOWNET with the active learning pipeline with three baselines. One is an active learning model as a generator of the sequences in a PPO. Another model is like more classic Bayesian optimization with an RL-based genetic algorithm and comps. You can check the references here. So these are, as you see, recent algorithms that are representative from the literature on sequence design. Yeah, and these are some results showing the metrics of that we discussed before: performance, diversity, and novelty. So one first conclusion is that GFLOWNET generates sequences with a score on par at least on par or even higher than the strongest baseline. In this case, the strongest baseline was Dyna PPO. So you see that the main advantage of GFLOWNET is not like dramatically improving on the average score of the sequences, but where we actually gain an important advantage with GFLOWNET is when we look at the diversity and the novelty of the sequence. This is the main motivation for using GFLOWNET. So you can see that even if the performance is similar, the other methods fall short at discovering truly diverse samples. And maybe to add to this, one reason that we observed is that methods like this, for example, COMS performs local search around known candidates. So it really exploits the knowledge from the original data set, but doesn't really generalize to unseen regions of the space, which is what GFLOWNET excels at doing. Okay, so this is what allows it to find diverse and novel samples. You may be wondering why this amortized BO Bayesian optimization is not in the table. So this also highlights one nice property, not unique of GFLOWNET, but one nice property of it. So in the case of amortized BPO, it is really designed for fixed length sequences, and in this case we didn't find meaningful sequences, so that's why we didn't include it in the table. Of course, GFLOWNET is flexible and it can generate sequences of various lengths. So to conclude the slides, GFLOWNET succeeded at generating sequences that satisfy all the three metrics simultaneously. And to conclude the presentation, we don't have time to discuss and present here all of the results that we have played with here, but we will mention it quickly. So the paper also includes results with DNA sequences that have high binding activity with human transcription factors. There is a data set and benchmark results on design of protein sequences with high fluorescence. And then we are currently working on other problems. So just to mention them, for example, well, actually the small molecules with high binding affinity with the target ligand. This is one of the benchmarks that was presented in the original GFLOWNET paper that Mox worked on. And we are currently applying GFLOWNET as well to find DNA aptasensors with low free energy and high binding activity with target ligand. This is also an application with high impact in multiple domains. Beyond biological sequence design, we're also using this for more inorganic material discovery with electrocatalysts, for energy storage, for example, or for improving batteries, searching for solid state ionic conductors. And other people in the group are working on the application of GFLOWNET on other domains. Something that is also included in the biological sequence design paper is the inclusion of epistemic uncertainty into the reward function. We found that this improves the training and the generation of diverse sequences, and we also explored how GFLOWNET training can be accelerated by including samples from the empirical distribution. So if this is something that interests you, you can see more details in the paper that's already on arXiv. And for future work, something that we are very interested in is in integrating GFLOWNET sampling for improving continual learning algorithms, and also exploring one active learning case that is quite interesting in the real world, which is exploring multi-fidelity oracles. And this kind of connects with the first slide where we were talking about how searching for new materials, for new sequences, for new proteins usually involves several steps. So if we can simulate these different steps with active learning and GFLOWNET as a sampler for multi-fidelity oracles, that will likely improve the current methods. And finally, to conclude the whole presentation, so we have talked about GFLOWNET, which is an effective method to sample objects from multiple modes of adaptive distribution. This gives us the property of sampling high reward, both high reward and diverse samples. This makes GFLOWNET a very suitable method for active learning pipelines. And we have give you some results on the specific case of biological sequence design. So like Joshua saying, GFLOWNET can, Joshua Benjo, our supervisor, saying, GFLOWNET can be regarded as a Swiss Army knife for probabilistic modeling. And we hope that these methods is developed further in the future, both here, but other groups too. So thank you very much, and we'll be happy to answer your questions.
Thank you.
Wonderful. Thanks, Alex and Moksh, for your presentation. It was very nice. So I think we can open the floor for questions. So for the participants, either you unmute and you ask the question directly, or you write it on the chat, and then I'll try to transfer the question to our to Alex and Moksh. So to maybe heat up a little bit, I would like to start asking, say, maybe a general question. So I have the feeling that this could also be applied to research in art and creativity. You could see each of these states as, one state of a painting or a sculpture. I don't know if you have any thoughts on that, if you have someone has proposed you to work on these directions or to research that.
Yeah, so actually that actually does make sense, in the sense that you can think of images as for example, you take the MNIST images, like the classic MNIST data set. There you can think of the images in the MNIST data set as a bunch of combination of a bunch of strokes put together. So you can have the stroke of a semicircle, or just a quarter of a circle as one of your actions, and the stroke of putting another semicircle as one of your action, as you just sort of piecewise put together these actions to construct your digits. So that is sort of something which is very feasible, and we have thought about it, and thought about doing this, but I haven't really gotten to doing that yet.
If I just can say a few words, I mean, it makes a lot of sense in that what really the way of thinking of GFLOWNET is something which is very important, and that's why I come back to this slide, is this idea of building blocks. So really objects that can be decomposed in different discrete episodes. Actually, it doesn't need to be discrete, but this is what we have focused so far. So objects that can be decomposed in discrete steps, and arts, sculptures, you said, imaging. I think it's a good example of that. We haven't really played with this idea, but it's quite appealing, actually.
All right, thanks. So maybe now that you are here in the theoretical part, there is something that wasn't completely clear to me. Is you mentioned that that you have these two policies, and I understand that this is what you have done in the laws, but I did not quite get the intuition on why do you need these two policies.
Yeah, so I apologize for that. We had to skip over a lot of things which are sort of motivate a lot of these things. So but just to briefly summarize, if you in the GFLOWNET Foundations paper, we have this another theorem, which is this detailed balance theorem, and this detailed balance or sort of the detailed balance matching condition. So it is very similar to what you would see in MCMC methods. So there is a detailed balance condition in MCMC methods as well, where you sort of look at the reverse probability of going backwards in the chain as well. So you have your forward you have your forward trajectory, and you have a backward trajectory. Now the reason why this backward policy is so there are two things. One is why it is useful, like sort of in a more practical way, and the other one would be why is it useful in a more like theoretical way. So in the more theoretical way, it is useful because essentially these flows, it sort of allows you to define your flow uniquely. So you can have like multiple flows which can satisfy this like you can have essentially infinitely many flows which can satisfy your flow matching condition. But in order to sort of make this forward flow unique, we introduce this backward policy, and for the sort of result is that for each backward policy you have a one uniquely defined forward policy. So that's the sort of the theoretical like sort of intuition for why we need this backward flow. A more practical reason would be, let's say you're given, like once you have sort of a trained GFLOWNET, now oh sorry, I just noticed there is a typo. It should be like a backward policy PB. Apologies for that. But yeah, so coming to the more like practical usefulness for this backward policy is like, given a particular molecule, let's say you have a fully constructed molecule, you can run this backward policy also to see what could be a potential, let's say, pathway of sort of generating this molecule. So this is not something that we have like actually verified in experiments or like try to do this, but if we can deconstruct a molecule with this backward policy, it will tell me, okay, I should first like the last step to be generated. So given a complete molecule, it will tell me, okay, the previous state it would be this. So essentially it would differ by one block. And like if we go up this pathway, we can sort of like get also a way to generate some given molecule.
Yeah, and just to add like very quickly to this, when you do that, because these flows can be seen as normalized probability distributions, then we can actually decompose the backward trajectories and define a distribution over the possible trajectories that could lead to this finished object, which is can be quite useful for analysis. Okay, so this gives you also a way to construct the object that you are sampling, right? Yeah. Okay. Okay. And you can, if the GFLOWNET is trained, you can actually see analyze the backward policy according to the trained GFLOWNET of any object. Doesn't need to be an object that you have used during training, for example. Yeah, I see. I understand that. Yeah. Okay. Good. All right. So if we can go maybe a bit more in details on the experiments that you guys have presented.
Yeah.
I was curious because so the way you have defined the novelty metric is something intrinsic to your data. So basically you check how far can you go, how different can you go. But of course it doesn't give you an idea on how much coverage are you having on the real outside, all the things you can do. And okay, I don't know if it's possible to have a measure, because if you had this measure, probably you don't have the problem anymore, right? But I don't know if you have any idea for the experiments that you did, how much of this coverage are you having? It's like, do you have any idea if it's small, it's huge? It depends on. The thing is, yeah, I guess it's possible to define metrics of that, but such a metric would really depend, or the usefulness of such a metric would really depend on the score of that coverage as well, because you can have a random policy and cover the whole, or maximize the coverage. But if that doesn't give you good performance, then this batch of samples wouldn't be very useful. Of course, we have no idea whether the top K batch is missing like good sequences in parts of the space that we haven't explored. We cannot know that. But given an average performance, then we can see what the novelty is, and that will tell us, well, these are good samples and they are also novel. I don't know if that makes sense, because you can have like very, very diverse and novel samples, but they could have like very, very low performance, and then this would be useful for practical applications.
Yes,
just-
I understand, but I'm just thinking about the initial figure when you were intuitively comparing to MCMC, and I am wondering, how do you know if you are jumping into the other mode, right?
Yeah, so that's a good question. So actually, like it's a bit tricky to like verify that in like these amps and like high dimensional cases. But we, in the original GFLOWNET paper, we do have experiments in a more toy setting, just to sort of ensure that we can actually compute like exact distributions and so on. So we have toy experiments where we basically have, let's say, 4 separated modes in the distribution, and like there is just zero probability mass in between, or like very low probability mass in between. And yeah, these are well separated modes, and there we sort of verify quantitatively and sort of qualitatively that we are able to cover all of these 4 modes, even when there is like very low probability mass in between. So when the separation is very high of these modes, we are able to sort of reach all of these, even the far away modes, even when we start from one of the particular modes. And MCMC methods that we compare to, they just sort of get stuck on this one mode, or like at most they're able to reach like 2 of the modes or something. So yeah, we do have like we don't have those results in this particular paper that we sort of covered mostly, but there are these results in the original GFLOWNET paper.
Okay, thanks. Yeah, that kind of answers my question. I mean, I wasn't expecting a completely full answer, because I don't know how to. I mean, I don't think it's possible to get it right. But
yeah.
All right, and then I have one last question, which is out of curiosity. Towards the end, you mentioned something that's epistemic uncertainty, and I am not familiar with that, so I would be very happy if.
Yeah, sure. So I guess basically when we just going back to Alex's diagram for the active learning setup. So the model which we train here, so this model which we have here, let's say this is like a neural network, right? We just train a neural network on this in a supervised fashion on some data that we have. Like, let's say your X is your sequence and Y is like, let's say, the antimicrobial activity score for this particular sequence, and we train a neural network. So one thing which, like all models, like no model is perfect, and like we will have some uncertainty over the predictions of this model. So this uncertainty can actually be very useful in sort of guiding the search of this particular agent. So in the sense that if we know that like my model is like, let's say we have some space where my model has seen a lot of data, it is very confident about. But as we move away from this training distribution, we are low and low, more and more less certain about the predictions of my model. So it might be useful to search in that area, which is like very high uncertainty. So there are ways, like lots of ways of capturing this uncertainty in like neural networks, which is not really like the focus of our talk, but like if you have some measure of this uncertainty in your model, it can be imperfect. Like most of the measures like which we have for uncertainty estimation of neural networks are imperfect, but even some information about how the uncertainty like how the uncertainty sort of evolves over the space, that can be helpful in sort of directing your search. And it's very intuitive in the sense that just to clarify, this is what we mean by the epistemic uncertainty, which is like the uncertainty in your model because you have like finite data sample. So yeah, it intuitively also makes sense that if there is a particular topic that I'm uncertain about, I'll try to read more about it. So like that's the intuition we have here. And it's also actually a very well studied thing in the context of like the literature of Bayesian optimization, where we have like a lot of these acquisition functions which sort of trade off like this exploration and exploitation, such as like the expected improvement, upper confidence bound, where you sort of weigh your uncertainty. So you add your like sort of uncertainty estimates to your like mean prediction, so that we account for the like imperfection in my model. So yeah, I hope that answers your question.
Yeah, it does. Thank you. Yeah, it does very well. Thanks a lot. All right, so I don't know if someone else wants to ask a question. We have no questions from the audience. I would say that this is because your presentation was very clear, because it was to me. I hope it was the same for the others. All right, so if there are no more questions, I would like to thank you again, Alex and Moksh, for your presentation. It was very interesting to have you here. And if we have any further question, we will not hesitate to send you an email and to ask for for more clarifications and how the hell can we start using these things.
Yeah, there's if you want, well, thank you for the invitation. It's been a pleasure for me, and thank you for that. If you want, we can actually send you some, well, the slides and also some other resources of how to get started with GFLOWNET. There's some
I guess we can add that to our webpage. So yes, we can put that if that's fine with you.
Yeah,
totally.
Yeah. Okay. Thanks a lot. So yes, we will be happy to have these materials.
Perfect.
Okay. So thanks once more, and I wish you a very nice day ahead. For us, it's the end, but you are just starting.
You have a good evening.
Yes. Okay. Thank you, guys.
Thank you so much.
Bye, everyone. Thanks.